Today, we will discuss on the topic membrane transport. We'll be focusing more on transport of metabolites across the plasma membrane. Let's look at the introduction first. Cell membranes are crucial to cell structure and function. The lipid bilayer of all cell membranes serves as a barrier to the passage of most polar molecules. This barrier function is crucially important as it allows a cell to maintain concentrations of solutes in its cytosol that are different from those in the extracellular fluid and in each of the intracellular membrane-bounded compartments. However, cells have also evolved ways of transferring specific water-soluble molecules across their membrane in order to ingest essential nutrients, excrete metabolic waste products, and regulate intracellular ion concentrations. This lecture will lead you through the important concepts of membrane transport so that you will understand its importance to living cells. We will also discuss the types of membrane proteins involved in transport of metabolites across the plasma membrane and the mechanism of membrane transport. All cellular membranes consist of a bilayer of phospholipids into which other lipids and specific types of proteins are embedded. It is this combination of lipids and proteins that give cellular membranes their distinctive permeability qualities. Phospholipid bilayers on their own are excellent chemical barriers, impermeable to virtually all ions, amino acids, sugars, and other water-soluble molecules. In fact, only a few gases and uncharged small water-soluble molecules can readily diffuse across a pure phospholipid bilayer. But cellular membranes must serve not only as barriers, they must also act as conduits, selectively transporting molecules and ions from one side of the membrane to the other. The transport of inorganic ions and small water-soluble organic molecules across the lipid bilayer is achieved by specialized transmembrane proteins. Each protein transports a particular class of molecules such as ions, sugars, or amino acids. The importance of membrane transport is indicated by the large number of genes in all organisms that code for transport protein, which make up between 15% to 30% of membrane proteins in all cells. Some specialized mammalian cells devote up to 2/3 of their total metabolic energy consumption to membrane transport processes. These membrane proteins act variously as shuttles, channels, or pumps for transporting molecules through a membrane's hydrophobic interior. The three main types of membrane proteins are ATP-powered pumps, channel proteins, and transporters. Now let's look at ATP-powered pumps first. These proteins are ATPases that use the energy of ATP hydrolysis to move ions and small molecules across a membrane against a chemical concentration gradient, an electric potential, or both, which is termed as electrochemical gradient. The overall reaction of ATP hydrolysis and the uphill movement of ions or small molecules is energetically favorable. Examples are the sodium-potassium pump, the hydrogen pump, calcium pump, etc. Now let's look into the channel proteins. Channel proteins transport water, specific ions, or hydrophilic small molecules across membranes down their concentration or electric potential gradients. Channels form a hydrophilic passageway across the membrane through which multiple water molecules or ions move simultaneously or in a single file at a very rapid rate. Some channels are open most of the time, and these are referred to as non-gated channels. Most ion channels, however, open only in response to specific chemical or electric signals, Now, these are referred to as gated channels because protein gate alternatively blocks the channel or moves out of the way to open the channel. Channel proteins, like all transport proteins, are very selective for the type of molecule or ion they transport. Examples are ion channels of sodium, potassium, calcium, chloride ions, etc. Now let's look into transporters. These are also called carriers or even sometimes permeases. Transporter proteins move a wide variety of ions and molecules across cell membranes, but at a much slower rate than channels. They bind a specific solute to be transported and undergo a series of conformational changes to transfer the bound solute across the membrane. They form continuous protein pathways across the lipid bilayer. Transport by carriers can either be active or passive. There are three types of transporters. There are uniporters, symporters, and antiporters. Now, uniporters transport a single type of molecule down its concentration gradient. Glucose and amino acids cross the plasma membrane into most mammalian cells with the help of uniporters. Collectively, channels and uniporters are sometimes called facilitated transporters, indicating a movement down a concentration or electrochemical gradient. One membrane transport protein found in the cells of liver functions to shuttle glucose between the liver and bloodstream and is an example of a uniport transporter because it moves only one solute. Next is symporters. They couple the movement of one type of molecule against its concentration gradient with the movement of one or more different ions down its concentration gradient in the same direction. A good example of a symport conformational transport protein is the sodium glucose transporter found in the renal epithelial cells of the kidney. This process is not passive because the potential energy of a steep sodium gradient is dissipated and used to drive the movement of glucose gradient against its concentration gradient. Last is antiporters. Antiporters couple the movement of one type of molecule against its concentration gradient with the movement of one or more different ions down its concentration gradient in the opposite direction. So, symporters and antiporters are often called co-transporters, referring to their ability to transport two or more different solutes simultaneously. However, the nature of energy supplying reaction driving active transport of these two classes of proteins differ. ATP pumps use energy from hydrolysis of ATP, whereas co-transporters use the energy stored in the electrochemical gradient. Co-transport process is sometimes referred to as secondary active transport. There is another class of molecules called ionophores, which are small, hydrophobic molecules found mostly in microorganisms that dissolve in lipid bilayers and increase their permeability to specific inorganic ions. There are two classes of ionophores, mobile ion carriers and channel formers. Both types operate by shielding the charge of the transported ion so that it can penetrate the hydrophobic interior of the lipid bilayer. Since ionophores are not coupled to energy sources, they permit net movement of ions only down their electrochemical gradients. Valinomycin is an example of a mobile ion carrier that transports potassium ions down its electrochemical gradient. Gramicidin A is an example of a channel-forming ionophore. Two gramicidin molecules are thought to come together end-to-end across a bilayer to form a transmembrane channel, which selectively allows monovalent cations to flow down their electrochemical gradients. It can transport about 20,000 cations per open channel each millisecond, which is about 1,000 times more ions than can be transported by a single mobile carrier molecule in the same time. Gramicidin is made by certain bacteria perhaps to kill other microorganisms by collapsing the hydrogen ion, sodium ion, and potassium ion gradients that are essential for cell survival, and it has been useful as an antibiotic as well. Now let's discuss the different mechanisms of membrane transport. Cells employ various transport mechanisms to selectively allow the movement of metabolites across its membranes. First, and the most simple type, is simple diffusion. Simple diffusion or free diffusion is the simplest type of transport mechanism. Small, non-polar hydrophobic molecules such as oxygen and carbon dioxide readily dissolve in lipid bilayers and therefore diffuse rapidly across them. Small, uncharged polar hydrophilic molecules such as water and urea also diffuse across a bilayer, but much more slowly. Free diffusion requires no energy expenditure on the part of the cell, nor does it require any transport carrier molecule. When the concentration gradient is dissipated, free diffusion stops. The rate of transport in free diffusion is strictly proportional to the concentration difference across the membrane. So the greater the concentration difference, the greater the rate of entry or exit. Now let's look at facilitated diffusion or transport. All channel proteins and many carrier proteins allow solutes to cross the membrane downhill by a type of membrane transport called passive transport or facilitated diffusion. In the case of transport of a single uncharged molecule, it is just the concentration gradient that drives the passive transport and determines its direction. If the solute carries a net charge, then both the concentration gradient and the electric gradient can be combined to calculate the net driving force, which is the electrochemical gradient of the solute. Therefore, in facilitated diffusion, the carrier or channel protein helps the molecule of the metabolite to be transported across a membrane along its concentration or electrochemical gradient. Here, the diffusion is facilitated since the molecule cannot penetrate the lipid bilayer of the plasma membrane on its own by free diffusion movements. Glucose uptake in animal cells, other than cells lining the intestines or kidney tubules, is achieved by facilitated diffusion. Cells can accumulate substances in excess and still create a downhill gradient in at least 3 ways. Number 1. The solute can be precipitated from solution once it is inside the cell so as to effectively reduce the concentration of the solute in the cytosol. The accumulation of calcium ions in the cisternae of endoplasmic reticulum operates by way of precipitation. Calcium phosphate is the precipitate in this case, and its formation maintains a favorable gradient of calcium ions. Number 2, the molecule can be chemically changed after it has gone through the membrane, thus reducing the concentration of the specific molecule involved in the concentration gradient. If sugar is phosphorylated immediately after entering a cell, then the inside concentration of the non-phosphorylated sugar remains low, and more molecules will be transported across the cell along the usual downhill direction of the concentration gradient. Number 3. The transport of a metabolite can be coupled directly to a second reaction that is energetically favorable to driving the transport reaction uphill. Now, let's look at active transport. Cells also require transport proteins that will actively pump certain metabolites across the membrane against their electrochemical gradient, and it is tightly coupled to a source of metabolic energy such as ATP hydrolysis or an ion gradient. This process is known as active transport and is mediated by carrier proteins, which are also called pumps. The process by which a carrier protein transfers a solid particle across a lipid bilayer, resembles an enzyme-substrate reaction, and in many ways, carriers behave like enzymes. But unlike enzyme-substrate reaction, the transported solute is not covalently modified by the carrier protein. Instead, it is delivered unchanged to the other side of the membrane. The active pumping of one substance out of a cell provides a driving force for the transport of various other substances inward. The pump is an economical and simple transport system since the outward movement of one kind of substance helps to drive in many kinds of metabolites. Cells carry out such active transport in three main ways. First, coupled carriers couple the uphill transport of one metabolite across a membrane to the downhill transport of another, for example, ion gradient driven. Secondly, ATP-driven pumps couple uphill transport to the hydrolysis of ATP. And third, light-driven pumps, which are found mainly in bacterial cells, couple the uphill transport to the input of energy from light, for example, bacteriorhodopsin in bacterial cells. The solutes that are most actively pumped into the cell are potassium ions, sugars, and amino acids. The driving force for this inward transport is sodium ion gradient across the membrane, created by the active transport of sodium ions pumped out of the cell. So we have come to the end of today's lecture. I would like to conclude by saying that the hydrophobicity of membranes is a barrier to the movement of hydrophilic compounds and ions into cells and organelles. To circumvent this problem, cells produce membrane transporter proteins that facilitate the movement of these lipid-insoluble molecules. Passive transporters accelerate diffusion, moving molecules across the membrane with their concentration gradient towards equilibrium. Active transporters use free energy to move molecules against equilibrium, generating a concentration gradient. Thus, several types of transporters function together to carry out important cell functions.